Hi, this is Dr. Neela Bakore. Meiosis was demonstrated by a scientist named Van Benden, and the term meiosis was given by another two scientists. Term was given by Farmer and Moore. So these two scientists gave the term meiosis, but the demonstration of this process, which was the reductional division, was given by Van Benden. Meiosis is also known as reductional division, and the reason for giving it this term is that chromosome number is reduced to half. Chromosome number is reduced to half. That means whatever is the number of chromosomes in the parent cells, in the daughter cells the number of those chromosomes would be half. At the end of meiosis, one parent cell gives rise to 4 daughter cells. One parent cell gives rise to 4 haploid daughter cells. The reason why we are using the term haploid, because the chromosome number is getting half. So a normal cell, which is a diploid cell, 2n number of chromosomes, has 2 sets. So the daughter cells will get half of it, that is a single set. That is why 4 daughter cells, one parent cell gives rise to 4 haploid daughter cells. Meiosis is completed in 2 major parts. One is known as meiosis 1, and this is followed by meiosis 2. The basic terms which we use for the phases, we have done this in mitosis, we talk of prophase, metaphase, anaphase and so on. Same terms will be used here, but to differentiate them from each other, we will write number 1 and number 2. So in case of meiosis 1, first is going to be karyokinesis. In this karyokinesis, same 4 subphases, that is prophase, metaphase, anaphase and telophase. So this will be written as prophase 1, then metaphase 1, anaphase 1. Names are same, only the numbering tells us that it is a part of meiosis 1. So if we write simple prophase, the indication is it is of mitosis, prophase 1 is of meiosis 1 and prophase 2 will be of meiosis 2. And the last one here is telophase 1. After this, the nuclear division will get completed and it will undergo cytoplasmic division, so cytokinesis. Same things will happen in this, but we know that before a cell divides, it prepares itself. That means here it has undergone that interphase, it has prepared itself for the DNA replication, the protein accumulation, ATP synthesis, everything is done in interphase, and now the cell is ready to divide. So we assume that here interphase has already taken place. Now this prophase 1 of meiosis 1 is again subdivided into 5 phases. These are leptotene, zygotine, pachytene, diplotene, and dikinesis. So these are 5 subphases of prophase 1. So the cell has prepared itself in interphase, then it undergoes karyokinesis. First phase is prophase 1, which is again divided into 5 parts. Then it will be metaphase 1, anaphase, telophase, and then cytokinesis. This will be followed by meiosis 2. Now, as a result of meiosis 1, there are 2 daughter cells formed. So, we will simply write it that here we will get 2 daughter cells. Now, both these daughter cells will undergo meiosis 2. So, these will enter this phase. Do they need to prepare, because this is also division, that is also division. So, do these cells require to prepare themselves? We have seen what all happens in this interphase. Gap 1, growth, accumulation of proteins. S phase, where DNA replication takes place. G2, where again the cells prepare themselves for the actual division. All that's happened is taken place in interphase. In this case, that means these 2 daughter cells which are formed, they do not undergo interphase. Instead, they undergo a phase which is known as interkinesis. Interkinesis has only growth part, that is accumulation of proteins, RNAs, everything will take place. Here in this interkinesis, there is no S phase. That means DNA replication takes place only once, and that is during interphase, not during interkinesis. So here there is no S phase. This will become more clear to us once we start explaining each and every stage and understand what changes take place. Now these 2 cells undergo meiosis 2. Meiosis 2 is again completed in 2 stages, that is karyokinesis, nuclear division. Again there are 4 phases, that is prophase. And how do we differentiate this from the prophase 1? We write it as prophase 2 followed by metaphase 2, then anaphase 2, telophase, and after this nuclear division has taken place, cytokinesis will take place. So we are using the same terms 3 times actually. Once during mitosis, second time during meiosis 1, and third time during meiosis 2. Easy to remember, in mitosis the words are simple words without the numbers written in front of them. Meiosis 1 we write 1 in front of all, meiosis 2 we write 2 in front of all. Should we not write 1 and 2 for this cytokinesis also? This is division of cytoplasm of meiosis 1, so cytokinesis 1, and this will be cytokinesis 2. As a result of this, now the division will be complete and we will get 4 daughter cells. When we start meiosis 1, or when a cell undergoes meiosis 1, the cell here is diploid. The cell is diploid. Then it undergoes interphase where it is going to make a copy of DNA. So it is actually 4n like genetic material wise or DNA content wise. When we come to this, the chromosome number is reduced to half. So here one cell, we will get 2 cells with n number of chromosomes, that is half the number. So meiosis 1 will be called reductional division. It is known as reductional division. These 2 cells which undergo interkinesis and now will pass through meiosis 2, we will get 4 cells. So 4 cells or 4 daughter cells are all haploid. That means after this, the chromosome number has not changed. It is similar to this. So this part of meiosis, that is meiosis 2, is equational. Mitosis is also equational. So meiosis 2 is similar to mitosis. Meiosis 1 is reductional division and meiosis 2 is equational division, which is pretty much same as mitosis. Now let us take all these stages one by one and see what all changes take place and how the chromosome number gets reduced to half. So now the details. Let us start with the first phase of meiosis 1, that is prophase 1, and we talked of that before the cell enters meiosis, it has gone through cell cycle interphase. That means this cell has already undergone G1, S and G2 phases. Now it is ready to divide. So the first cell that we are drawing is a cell after interphase, and we have already talked about this, that the cell after interphase has a large nucleus and again we are taking an animal cell, so we will draw a centrosome. So let us call this cell interphase cell. Now it enters the process of meiosis. It will go through karyokinesis of meiosis 1, and the first is prophase 1. We said that prophase 1 doesn't take place in one stage, it is completed in 5. So we are talking about these 5 sub-phases of prophase 1. The first is leptotene. This phase is also known as leptonema phase. Leptonema, leptotene or leptonema. In this, the nuclear membrane remains as it is, that means, and keep comparing it with what we did in case of mitosis. In prophase of mitosis, this nuclear membrane disappeared. Here also it will by the end of prophase. That means once we do all these sub-phases, towards the end it will disappear, but in this one it is intact. Centrosome, that is the clear cytoplasmic area and the 9 peritubular fibrils, they would disappear, and this centriole, it divides, and we start seeing aster fibers. One centriole remains at one pole, the other will start moving towards the other pole. The chromatin fibers, and again for our understanding we are going to take a fixed number of fibers so that we are able to keep a track of all those. These fibers 1, 2, 3 and 4. Let us talk about 4 fibers. let me draw these 1, 2, 3, 4 here, and after the changes here. Now these chromatin fibers, they start to condense by dehydration. They start losing water, start becoming thicker, but this thickening is not uniform. The thickening takes place at intervals. So we see the fiber showing intermediate thickenings. It is undergoing condensation, but instead of a uniform thickening which would show us the typical chromosome shape, we are seeing thickenings at intervals. And as you can see in this diagram, all these chromatin fibers at one end are converging. They are coming towards one side, and the other end they are diverging. So it appears as if it's a bouquet of flowers. So when we make a bouquet, the upper end is wider and the twigs or the stem part is tied together. So same thing, this part is narrower and the upper part is spread out, and that is why this stage is also known as bouquet stage. Bouquet stage, just because of the appearance. The intermediate thickening is because of incomplete or random thickening or dehydration. It will become completely condensed and we will start seeing the shape of the chromosome very soon. This is leptotin stage. Nuclear membrane as it is, the nucleolus which is inside the nucleus, that also remains. Centriole splits, one at one end, the other centriole starts moving towards the other end. Aster fibers appear. Now this cell enters the next subphase. The next subphase is known as zygotene or zygonema. Nuclear membrane is still intact. Centriole, one is at one end, the other is almost at the other end with their aster fibers. The chromatin fibers have condensed further, and here we start seeing the chromosomes. Not very thick, but chromosomes are. And these chromosomes, they arrange in the form of homologous pair. Now here we need to understand what exactly we mean by homologous pair. Let us go back to the original cell. This cell, interphase cell, is a diploid cell. That means it has two sets of chromosomes. If you are talking about human cell, 23 chromosomes are from the mother and 23 chromosomes are from the father. That means for one set of characteristics, there are two chromosomes. Suppose you are talking of eye color. So there is one chromosome coming from mother with the eye color gene, and the second chromosome is coming from the father, but for the eye color gene. So for eye color gene, there are two chromosomes. So for everything, there are two chromosomes. This pair of chromosomes is known as homologous pair. So if there are 4 chromatin fibers, 4 chromosomes, there would be 2 homologous pairs. So now these chromosomes, they arrange in the form of a homologous pair, and we will use 2 colors to show these 2 pairs. Homologous pairs. What are homologous pairs? A pair of chromosomes responsible for the same characteristic feature. Out of this pair, one received from the female parent and other received is from the male parent. So if there are 4 fibers or chromatin fibers, half the number would be of the homologous pairs. One pair or every pair is actually known as a bivalent. This pair is called a bivalent. So how many bivalents are visible here? 2, because we started with 4 chromatin fibers. Zygotene stage. in this zygotine stage, we also start seeing some spindle fibers, but the spindle apparatus is not clearly seen because the nuclear membrane is as it is. Now this enters the next subphase. Next subphase is pachytene. Many important things take place in this phase, in pachytene. These chromosome pairs, they will undergo vertical splitting. That means, I am going to draw a separate stage. Say this is one pair of chromosomes. So this is one pair. This pair splits vertically. Assume this is one arm and it is going to split like this. So now you have two arms here. So this will be seen as one chromosome having two arms and other chromosome also having two arms. We have given one term here. This term is for a pair of chromosomes which is called bivalent. Here, one out of the pair, one chromosome we are talking of, is having two chromatids. These two chromatids are of the same chromosomes, so these are known as sister chromatids. Sister chromatids. And this structure, we were calling this part as bivalent. One chromosome of the pair or of a bivalent, when splits vertically, the structure which we see is known as a dyad. And when you talk of this whole thing, two dyads together, then it is known as a tetrad. Clear? One pair of chromosomes, one pair of homologous chromosomes is known as bivalent. Each chromosome of this pair undergoes vertical splitting. So each chromosome has two branches and these branches are known as sister chromatids. One chromosome with two sister chromatids is known as a dyad. There are both chromosomes undergoing the same kind of change. So now we see the pair undergoing vertical splitting. So each one with sister chromatids. This complete structure is known as a tetrad. Now what will we call a branch or a chromatid of one chromosome and a chromatid of another chromosome? The two chromatids of one chromosome, sister chromatids. These two chromatids of other chromosome, sister chromatids. But when you talk of this arm and this arm, these are the chromatids of two chromosomes. They are known as non-sister chromatids. This is important for us to understand because crossing over takes place between non-sister chromatids. It cannot take place between sister chromatids. Okay. So now if we have to just compare that, suppose we started with two chromosomes, one and the other one, and I am showing just one half of it. Assume this is the centromere and this is the branch or the chromatid. When it splits, it appears like this. These two are sister chromatids and these are also sister chromatids. But if you are talking of one and one of this, they will cross over. So crossing over is possible between non-sister chromatids of the homologous pair. Now in this pachytene stage, this crossing over takes place, but before that, when these chromosomes, they come together, they must remain together. It is all in the nucleoplasm. There are all chances that these two chromosomes come together and again move apart. So there has to be something which is going to hold these two things together. We need not draw this, but there are some nucleoprotein fibers which are formed between them. So there would be some structures like this which are formed between the two members or two chromosomes of the homologous pair so that they remain together. So in this zygotine stage, we also say that there is one more thing which has appeared are these connections. These are connections of nucleoprotein fibers between the chromosomes of the homologous pair. This is known as synaptonemal complex. So synaptonemal complex, we are highlighting the events which are taking place in all subphases. So synaptonemal complex is the connection between the chromosomes of homologous pairs. These are fibers made up of nucleoproteins, and they would help the two chromosomes of the homologous pair stay together. That is when zygotine stage is taking place. This is called synaptonemal complex. What happens in pachytene? Now these two chromosomes of the homologous pair are together. Why are they together? They are together because of this synaptonemal complex, and they will undergo vertical splitting. So here what we start seeing in this stage is a tetrad. Why we made all these things separately? Because here we won't be able to show all changes in one cell. So these are the sequential changes, and here we will draw the end result or the final things. Nuclear membrane starts to disappear. So you can make it in the form of a lighter structure. It is not going to disappear. Again, we are using the word disappear, but it is not a magical disappearance. These nuclear membrane fragments break up and they join ER. So basically it dissolves back into the cytoplasm. But the more common term used to explain this is disappearing only. So nuclear membrane starts to disappear and we start seeing tetrads. How many tetrads are we going to see for one pair? One tetrad. So we are talking of one chromosome, its homologous pair. This is one pair and it's dyad and dyad. So together this is a tetrad. Similarly, we have one more pair. So now here, because we started with 4 chromosomes, we are seeing these 2 tetrads. Towards the end of pachytene, one more change takes place. It is known as chiasma formation. So in pachytene, chiasma formation takes place. What is chiasma? Chiasma is a cross between the non-sister chromatids. So if again we are talking about this, a cross takes place. It is represented by English alphabet X because this crossing over is taking place. So let us show this cross at one place, or let me just show it to you at separately here, and then we will put that same change in the diagram. So now if crossing over is taking place, then how will it look? This is one chromosome and its chromatids. This is its homologous pair and its chromatids. Suppose crossing over is taking place between these 2 non-sister chromatids. So these non-sister chromatids, they will overlap. They will make a cross. So here we start seeing a crossover, and that junction is known as chiasma or chiasmata. So this point is chiasma. What happens in this? The theories, there are multiple theories, but the theory which is most accepted one is that these fragments, they fuse, break and get exchanged. That means after this what is going to happen is that a part of blue chromosome which we have drawn here, this sister chromatid showed crossing over with the pink one which we are showing here. So at the junction, these two arms will fuse first and they will break. Break in the sense that one part, the part of pink goes with blue and part of blue goes with pink. So after they separate, it will be a part of pink coming here and a part of blue going here. What has happened because of this? Because of this, genetic material of one sister chromatid has been exchanged with the genetic material of the other chromatid of the other chromosome of the same homologous pair. This is called crossing over. Crossing over, chiasma formation takes place towards the end of pachytene. Crossing over, that means the complete fusion and exchange. It starts either at the end of pachytene or beginning of the next stage. In the first case, when we were talking of mitosis, we said there is no clear demarcation, that one phase ends here and the other starts. So there is a little overlap. So crossing over starts either in the late pachytene phase or in early next phase, that is diplotin phase. So we are showing the crossing over or chiasma formation in this case. So we will show crossing of, this is the blue arm and there is X shaped structure formed which is chiasma. So let us label this thing as chiasma or chiasmata. How many such chiasma can be seen? Multiple, depending upon the length of the chromatids. Upper arms can also cross over, number of chromosomes. So every arm can cross over, but crossing has to be between non-sister chromatids only. Now this cell, okay, let us draw this centriole also, which has reached at the two poles and with their aster fibers. Once this nuclear membrane is completely gone, we will start seeing the spindle fibers again. Now the cell enters the next subphase. This subphase after pachytene is known as diplotin. It is called diplotin. In this, nuclear membrane is completely gone, so we don't see nuclear membrane. We will have two centrioles with their aster fibers and spindle fibers will also be seen, but we are not drawing spindle fibers because we have to show this chromosome thing which is more important, and then later we will draw the fibers. Chiasma formation takes place in pachytene. Crossing over either starts at the late part of pachytene or here. So we will show this chiasma again where crossing over is taking place. Let us take the same chromosomes, the blue one here and the yellow one where one arm of yellow, they are crossing over, and the other one is, and we have not shown this here, but crossing over can take place in these chromosomes also. So in diplotin, this crossing over gets completed. So here the important event which is taking place is crossing over. Crossing over is beginning somewhere in the end of patchitine and getting completed in this diplotin, or it can start in early diplotin and get over by the end of it. Again, what happens in this crossing over is the two crossed arms of non-sister chromatids, they will fuse at this junction of X, and then the upper parts after X they would get exchanged. So now after this we will see these parts exchanged, and just to make it a little more prominent, let us make these arms solid so that we can see it clearly. Here crossing over gets completed. Now when crossing over is done, the easiest way is that they should separate like this. They fuse like this, they separate like this. It is not like this. They fuse and the separation is like this. So if you can see these two, the separation is slowly they come towards the end and separate. It's like as if we are opening a zip. So when the zip opens, it opens gradually like this. So this X also separates these two arms, they come closer to the end and separate. It is known as terminalization.